Good morning, everyone. Today we are going to study one of the most fundamental topics of microbiology, pure culture, and its isolation techniques. Before we understand techniques, first we must understand why pure culture is needed. Because in real life, microorganisms never exist alone. They always live as a mixed population in soil, water, air, food, even inside our body. So, the main question is how can we study one microorganism when so many are present together? The answer is pure culture. What is pure culture? A pure culture is a population of microorganisms that contains only a single species obtained from a mixed culture. In simple words, I can say that mixed culture means many microbes together. Pure culture means only one type of microbe. This concept was first developed by Robert Koch. Robert Koch, है ना? A German bacteriologist. He is logic. Identify a microorganism, study its structure, test antibiotics. We must isolate it first. Need of pure culture, importance of pure culture. So now I will explain why pure culture is required point by point with reason, not just a theory. First, to identify and study the characteristics of microbes. So identification is impossible in mixed culture. So identification is impossible in mixed culture because different microbes have a different shape. Different microbes have a different shapes, different metabolic activities, different staining reaction. In mixed culture, everything gets confused. Pure culture, clear identity. Second point is to detect causative agent of particular disease. Suppose a patient has an infection. Suppose a patient has an infection. Sample contain pathogenic pathogenic bacteria and non-pathogenic bacteria. If we do not isolate the pathogen, we cannot confirm the disease. That is why Koch postulates required for a pure culture. For detect causative agent, we need to isolate the microbe. Third point is in order to find out antibiotic sensitivity and bacteriophage and bacteria susceptibility means antibiotics act as a specifically. If a culture is mixed, one bacterium may be sensitive, other may be resistant. Pure culture gives accurate antibiotic sensitivity results. Fourth, mutation rate in a pure culture is low. Clone is 99% identical. In pure culture, all cell comes from a single parent. Genetic variation is very low. That is why clone clone means progeny is 99% identical. And this is important for research, vaccine preparation, industrial microbiology. कंसेप्ट ऑफ CFU यानी कि CFU की फुल फॉर्म है क्लोनिंग फॉर्मिंग यूनिट. A single cell from pure culture give rise to a single colony. That colony is called CFU, colony forming unit. This concept is used in microbial counting, food microbiology, and water testing. 6. To obtain sufficient growth for the preparation of antigen and for other tests. To prepare vaccine, antigens, and enzyme, we need a large amount of a same organism. That is only possible with pure culture. To maintain stock culture for future testing. Pure cultures are preserved for a future research, teaching labs, industrial use. Without purity, results will be reproducible. Now, the isolation techniques of a pure culture. Now comes the most important part. How do we obtain a pure culture from a mixed culture? First is streak plate method, second pour plate method, third spread plate method, fifth enrichment culture method, lone culture, stalk culture, step culture, serial dilution method, single cell isolation method. So first we streak plate technique. The streak plating technique isolates individual bacterial cell of cloning forming unit on the surface of agar plate. This is most common used method. Gradual dilution of microbes on agar surface. First thing, mechanical separation using inoculating loop. We strike the culture in a different direction. Each strike reduces the number of cells. Finally, single cell gets separated from isolated colonies. Routine lab work or clinical sample. That's in any direction we separate or isolated. So for that we have to do staking is a process of spreading of microbial culture with inoculating needle or loop. Inoculating needle of loop means this. This is used for a separation of bacteria. And it is made up of platinum wire and nichrome wire on the surface of media. स्टेरिलाइज करना होता है इनोक्यूलेटिंग लूप को विथ अंडर फ्लेम टिल रेड हॉट, जब तक वो लाल कलर की ना हो जाए तब तक, एंड अलाउ इट कूल फॉर 20 सेकेंड्स. ठीक है. देन मिक्स्ड कल्चर में से इनोक्यूलेटिंग लूप से लेके थोड़ा सा हिस्सा निकालना होता है और अगार की प्लेट पे रख के उसको स्टिक करना होता है, यानी कि इस डायरेक्शन में ऐसे या फिर ऐसे कोई भी डायरेक्शन में कर सकते हैं. इससे हमें पता चल जाएगा सिंगल माइक्रोबियल की प्रेजेंटेशन और प्रोजेनिंग कैसी है. रीस्टैक कर सकते हैं वापस से फ्रेश मीडिया टू इंश्योर प्योरिटी. वापस चेक करने के लिए प्योर है या नहीं तो वो भी चेक कर सकते हैं. तो थैंक यू सो मच.